everyone. I think we can start. Welcome, everybody. I know that some people may be still logging in, but we'll make some introductions. Welcome to our webinar in collaboration between Pfizer and the MDA to present the CIFFREO clinical trial. Our speaker today, Kasia Lobello, will be speaking in a minute. I'm Nicolas Garnier. I'm Senior Director of Patient Advocacy at Pfizer Rare Disease Global Product Development and supporting this trial in collaboration with the Duchenne community. And very happy to introduce my dear colleague, Kasia. Kasia Lobello is the Global Clinical Lead, as I just said, for Pfizer Duchenne Muscular Dystrophy Gene Therapy Program. She received her undergraduate training in neurobiology from Cornell University, followed by a medical degree from the University at Buffalo School of Medicine and Biomedical Sciences. She completed residency training in neurology at Duke University, and since joining Pfizer in 2007, Kasia has worked to advance the treatment of neurologic and psychiatric disorders, including Alzheimer's disease, epilepsy, depression, and of course, neuromuscular conditions. Again, welcome. Please, if you have any questions, feel free to write them in the chat, and hopefully we'll have a few minutes at the end of the presentation to answer some of those questions. Kasia, over to you.
Thank you, Nicola, for the introduction, and thanks everyone for joining today. So I'm going to give you a brief overview of our phase 3 study in ambulatory boys with Duchenne muscular dystrophy. And as you can see, it's a double-blind, placebo-controlled trial. And we're looking at safety and efficacy of mini-dystrophin gene therapy. And you might also see it referred to as fordadistrogene movaparvovec. And this slide just shows some of my disclosures. And I thought I would start with an overview of gene therapy. So on a very basic level, I think we all understand that there is an abnormality in the gene that causes dystrophin, and that this is the underlying issue in Duchenne muscular dystrophy. And gene therapy is really a gene replacement treatment. So our aim is to take a copy of a working gene and then to give it back to the patient. In our construct, the gene, which is made up of DNA, is packaged into a viral vector, and the vector that we use is called AAV9, or adeno-associated virus number 9. And that gene that's in that viral vector is administered intravenously to patients and expressed, or it's taken up through the blood and then to various tissues in the body. It's expressed specifically in 2 target tissues, and so for Duchenne, those are skeletal muscle and cardiac muscle, or the heart. So this next slide is just a cartoon of what our mini-dystrophin looks like, and at the top you see there in red that is meant to represent the DNA portion of the gene. And then you see that flanking the gene on both sides are a series of promoters and enhancers. And again, that really ensures that the protein itself is manufactured in the tissues that we want it to be manufactured in. And then on the left in purple, you see sort of a more creative representation of what this viral capsid looks like. And then the bottom cartoons show you working down full-length dystrophin and what that looks like, followed by dystrophin in patients with Becker muscular dystrophy, and then followed by a cartoon depiction of our mini-dystrophin construct. So full-length dystrophin is really big. In fact, it's the largest human gene, and it's over 11,000 base pairs. And because of that size, we're not able to fit that full gene into this viral capsid that we would need to fit it into in order to deliver the gene to patients. And that's why what you see at the very bottom, our mini-dystrophin, is missing a big chunk of DNA. And the reason that we have confidence, one of the reasons we have confidence that this mini-dystrophin will provide efficacy in patients with Duchenne is that it closely mimics the dystrophin that's found in patients with Becker. So we know that Becker muscular dystrophy is a milder phenotype. Patients have the ability to ambulate for much longer time periods, for example, than people with Duchenne. And the key sort of functional parts of the gene on both sides are preserved and provide support for the muscle, even though it's not the full-length gene. So that was a little bit about gene therapy. And then I want to take some time to tell you about our study. It's called the CIFFREO Study. And at the bottom, you see a depiction of sort of the main three parts of the trial: the main study, the delayed treatment period, and the long-term follow-up. And you can also see that we're aiming to enroll approximately 100 patients. And in the main study, patients are randomized into either cohort 1 or cohort 2. Those participants who are in cohort 1 will receive gene therapy treatment right after randomization, whereas participants who are in cohort 2 will receive placebo. And in the main study, participants will be followed for 12 months, and this is where we will do our comparison between drug and placebo, looking at efficacy. And our primary efficacy endpoint is change from baseline at week 52 in the North Star Ambulatory Assessment Score. After the main study portion, there's a delayed treatment portion of the trial. And here, those participants who initially received placebo will now have the opportunity to receive gene therapy, assuming they still meet eligibility criteria. And then in the long-term follow-up, we will continue to follow patients for safety as well as efficacy for 3 to 4 years, depending on how long it's been since they received the gene therapy. And I'll go into additional details on the study in the coming slides. So a bit more about the design. So really the key sort of part of the study is 2 years in length. And again, at randomization, about 2 out of 3 participants will be randomized to active compound and will receive gene therapy right away. And about 1 in 3 participants will be randomized to placebo. One thing that we ask is that the child remain on a stable dose of steroids for the 2 years, for this 2-year portion of the trial. And the reason that we do that is we understand that steroids can change functional assessments and can sort of interfere with our efficacy signal. And so any changes that we see in terms of what the boys can and cannot do, we'd like to be able to attribute to our therapy and not have that confounded by any changes in steroids that the boys might have received. Additional details about the trial, there is a screening period and that can take up to 3 months. And during this time, the potential participants will undergo testing to make sure that they meet criteria for the study and to make sure that this is the right study for them to participate in. As I mentioned, there's a long-term follow-up period that can last up to 4 years, and that is a long time. However, during that long-term follow-up, we don't have as many visits and as many tests that are done, and so usually participants will only come in every 6 months or so. And again, one point I want to emphasize one more time is the idea of delayed dosing. So although there is a placebo arm, any participant who receives placebo and then who remains in the study and meets criteria after 1 year will be eligible to receive gene therapy. So that potentially, all of the participants would eventually receive gene therapy in the trial. A little bit about the clinical visit information. So it is an intensive study in terms of the number of clinic visits that are required. So we estimate about 49 total visits over the course of the entire study. And one thing that's unique about CIFFREO is that after dosing, we do require that the child stays overnight, that is, stays as an inpatient for at least 1 week. And we do this to ensure very close monitoring of how the boy is doing, both clinically and in terms of laboratory and other tests. So again, there's an inpatient stay of at least 7 days. And then after that inpatient stay, we ask the child and the family to remain close to the site for at least another week until we are confident that that they're doing well clinically. Most of the other planned visits are outpatient, and most of the visits take place in the first 2 years of the trial. So again, the first 2 years is where we do sort of the most intensive assessments of the boys. And again, the clinic visits are really important to us because they give us the opportunity to assess the child's overall health and well-being, see how they're responding to the drug. So this is just another cartoon of the visit schedule, and you can see that in the main study, you have the screening period, and then the baseline, and then the dosing. And in the first 2 weeks after dosing, you stay in the hospital for a week, and then you might need to visit the clinic every 1 to 2 days. And that's true whether you receive placebo or whether you receive the study drug. So in our delayed treatment portion of the trial, the visits parallel what you see in the main study. After that initial period, through month 3 of the trial, visits are every 1 to 2 weeks. And then for the 9 months after that, visits are every 1 to 3 months. And then for the long-term follow-up, as I mentioned, visits are every 6 months or so. Now, we had a question that we received before this WebEx about cardiac monitoring specifically and what kind of cardiac monitoring we do. So I thought I would just take a minute to address that while I'm on this slide. So cardiac monitoring really starts at screening in that we have a requirement for a pediatric cardiologist to assess any potential participant for eligibility in the trial. And then because the child is in the hospital for the first week, we have very intense cardiac monitoring while that child is in the hospital. And that includes laboratory assessments of measures such as troponin, cardiac troponin, as well as echocardiograms and other assessments as needed. And then that monitoring continues throughout the trial, although on a less frequent basis. We also have an algorithm in our protocol that directs study teams as to what needs to be done in case the patient develops some abnormality. So for example, if there's a rise in their cardiac troponin, we have an algorithm as to what needs to be done, and that involves consulting cardiology early on. And we also have a requirement in our study that any site that participates in the study has to be capable of providing intensive care if needed. So for example, if the participant needed any kind of mechanical support, we make sure that that's available and in place prior to any subject participating in the trial. So that's our cardiac monitoring plan in the trial. Okay, so I wanted to talk a bit more about who's actually eligible for the study. And you see the age requirement there. So boys aged between 4 and 7 years at screening are eligible. That is up until their 8th birthday, they are eligible. They have to have a confirmed diagnosis of Duchenne muscular dystrophy, and that's usually done by prior clinical genetic testing. And they have to be ambulatory. So our phase 3 trial is in ambulatory boys with Duchenne. And I mentioned earlier that we ask that the boys are on stable courses of steroids throughout the study. We also ask that they are on stable doses of glucocorticoids for at least 3 months prior to screening. And again, that is just to ensure that any changes that we see are due to our treatment and not due to any changes that they're having in their steroid dose. So what are some of the key exclusion criteria? So one obvious one is that if patients have an antibody to the virus or to our AAV9 capsid, they at this time cannot be included in the trial. And we do that, for safety and efficacy reasons, because those antibodies would neutralize that virus and we wouldn't get the desired effect. So that's a test that's done at screening. We also, as you might expect, do a number of laboratory tests during the screening period, and some examples are listed there. And significant abnormalities could exclude a boy from participation. And we also ask that boys who enroll in our study have not been on any treatment that is designed to increase dystrophin within 6 months of screening. And some of those treatments are listed there as well. So a couple more things while I have this slide up. This is just where you could go to get more information on our trial. And one point that I forgot to make on a previous slide is just that there's financial compensation available to families given the intensity of the study. So we can definitely discuss that with patients, or at least the sites can discuss that with patients, because we do understand that it's a very time and resource intensive study. And I also wanted to address one other exclusion criterion that I forgot to mention before. So sorry. So another exclusion criterion that we've implemented is to exclude boys who have a deletion in exons 9 through 13 or any abnormality in exons 29 through 30. So those boys with those specific mutations are currently excluded from participation in the trial, and that's really being done for safety reasons. So we also had a question as to whether or not we could talk to some of the side effects or the adverse events that we've seen in the study so far. And one thing that we have seen is we've seen some cases of cardiac inflammation and peripheral muscle inflammation in those boys who have those exons that I just mentioned. And we think that that's an immune reaction to the dystrophin protein, and that boys with those specific mutations are at a risk for that reaction. And so that's why we exclude boys with those mutations. And then just to talk about some of the other safety findings that we've had in the program. So probably the most common adverse event that we see is nausea and vomiting, and that's really seen in the majority of patients over the first few days to a week after dosing. And it usually resolves in a few days, and having the boys in the hospital where they can be given intravenous fluids if needed, and also given antiemetic medications is really helpful in dealing with the nausea and vomiting. Some of the other adverse events that we have seen, we believe, are due to an immune response to the virus, and these can include things like thrombotic microangiopathy. So we've had cases where there's been a decrease in platelets and some renal function abnormalities also. And again, we have a good understanding now of how to monitor and minimize the risk of those adverse events, but those are adverse events that we've seen. And then last, I did want to note that we did have a participant who unfortunately died last year after participation in our phase 1 trial. So we had a participant who died within about 7 days of receiving gene therapy. It was an unblinded phase 1 trial, so we know he received gene therapy. And just to say that he was 16 years old, so he was significantly older than the boys that we're enrolling in this trial, in the phase 3 trial. And like many 16-year-olds with Duchenne, he had pretty significant cardiac abnormalities at baseline. And so we think that there might have been that might have contributed to the reaction that he had. So that's a little bit about the safety profile. of for our program. And let's see. Also just wanted to thank everyone, especially for giving us the opportunity to present today and to present our program. And I think that's all I had. Nicola, did I forget anything? And also, are there any other questions?
Yeah, no, thank you so much, Kasia. I think you covered everything. So I'm not seeing any questions right now in the chat, but maybe we can give it a few minutes to see if anybody in the audience has any questions.
Okay, and if folks don't have questions now, I'm sure they can. Did you just get one, Nicola?
Yes, I
Okay,
see
go ahead.
someone is typing.
Okay, oh, somebody's typing. Okay.
Suspense. All right, so there's a question. We'll try to. Do we know the risk for boys in the placebo who may be exposed to the vector?
Yeah. So I think the risk is that. Well, no. So I think in general, if a boy were to receive placebo, he would not be exposed to the vector, right? He would receive placebo. There would be no vector administered. So there wouldn't be any risk of exposure to the vector. There's no vector in our placebo. So that boy's chances, if that boy is negative for antibodies at screening, the only risk is now he has to wait another year and then screen negative again before he's eligible for the dosing of the gene therapy. But receiving placebo will not increase his risk in any way of becoming antibody positive.
So follow-up question, is there no shedding from those who received the drug? And maybe you can talk a little bit about the sibling protocol.
So that boy who receives placebo, should not be exposed to other boys who receive the drug unless there's a sibling, right? So we do have a sibling protocol. So if there's two boys, two siblings, what we do is we ensure that they're randomized to the same arm. So if one boy receives placebo, we will make sure the other boy receives placebo alongside. And then in 1 year, they're both now eligible to receive study drug. If they both are if one boy is randomized to receive study drug initially, we make sure that his brother also receives the gene therapy. And we have to be careful in how we treat that in analysis. Only one of those boys ends up being analyzed, but for purpose of the family, both boys will be treated in the same way to guard against sort of being exposed to a sibling and then becoming positive and becoming yourself ineligible. I hope that helps clarify. It's a good question.
Thank you.
I don't know if the person is typing, if
Yeah,
that clear. Okay.
they're typing. In the meantime, someone else is asking, can you remind when does enrollment begin? And yeah.
Sure. So I think they're asking, so enrollment in the phase 3 trial has already begun, so we're already enrolling patients. And I think the question everyone wants to know is when will we start enrolling patients in the United States? And I can tell you that we are literally hours to days away from being able to tell you that we've had a site open in the US. So that will be posted on clinicaltrials.gov when it happens, and it literally like could happen today or tomorrow. So we're literally just days away.
Thank you. And we're all excited about that. Other question, can you remind how often will CK level be measured?
I'd have to look in the protocol to see exactly how often CK levels will be measured. We don't measure CK too often because when we're monitoring heart safety, the CK can change from the skeletal muscle, and so it's not necessarily super informative. So we do check CK, but we're really more focused on things like troponin when we're monitoring for heart safety, and that's initially measured daily right after the dose.
Excellent. Maybe we can give a few more seconds to see if anybody has another question. But in the meantime, I did drop my email address in the chat if anybody wants to write it down and think about any questions subsequently, and then I can make sure to direct questions to Kasia as needed. We got something else coming. So I guess build up on that question about the CK levels. Along with the troponin, are you monitoring the cardiac CK levels?
So we are monitoring CK levels, absolutely. Again, it's just not something that we're sort of prioritizing in our safety monitoring for the reasons that I discussed. But yes, we are checking CK levels.
There's a follow-up coming.
Awkward over the typing.
While the person is typing, I also want to remind that there is a website, CIFFREODuchenneTrial.com, that is available with a lot of information out there. So potentially, if people want to go and have a look. All right, so I don't see that any other questions coming in. So again, my email is in the chat. Obviously, you can also reach out to the MDA who can redirect questions to us. Thank you, Kasia, for presenting the study and answering the questions. Again, thank you to the MDA for hosting this webinar, and thank you everybody for listening.
Thank you so much. Take care. Bye-bye.
Bye.